Our final speaker in this session is Dr. Brandon Lind, who's a research fellow, studies the genomics of climate adaptation and biodiversity informatics at the University of Connecticut. His expertise is in adaptation and climate vulnerability, ecological genomics, and quantitative traits. His talk today is titled Branching Out, Evaluating the Use of Genomics to Predict Responses of Plant Populations to Climate Change.
Hi, my name is Brandon Lind. I'm a postdoc at the University of Connecticut with Joe Wegerson, but today I'll be talking about some of the work I've done at a previous postdoc with Katie Lauderhouse at Northeastern University. As we all know, the next century is forecasted to bring increasingly novel climates to many ecosystems across the globe. This is data from a paper from 2017 that shows climate novelty across North America. But what I mean by climate novelty is the multivariate climate that with no analog in the location's previous history. So of course this is going to create biological challenges, particularly for species that are locally adapted to climate. I study forest trees, and so as an example, without any climate-aware intervention, forest productivity could severely decline, seeing decreases upwards of 50% in California alone. So this type of information that is used to make this forecast come from reciprocal transplant designs, which are often resource and time intensive, particularly for long-lived organisms like forest trees. And so over the past decade, people have turned to genomic data to model relationships with climate of natural populations, and then to predict their vulnerability from climate data, which can come in hand much sooner than these transplant experiments and inferences from them. These genomic methods that I mentioned are generally referred to as genomic offset methods, and ultimately they ask the question, what is the genetic change necessary for a population to maintain climate adaptation? These models are created by sampling genetic and environmental data, multivariate environmental data from the landscape, and there's all sorts of different kinds of offset methods, but each of them create a model that relate genetics with climate. And then for a given population, we know its current genetic composition and its current environment. And given some forecast of climate change, we can use that model to estimate the necessary genetic composition of that population in the future. And the change in that composition is what is generally referred to as genomic offset. So a large genomic offset means that population will need to change quite a bit to maintain that relationship, which is increasingly unlikely as that genomic offset increases, meaning that that population could be more vulnerable. And of course the opposite is true as well. If there's little genetic change needed, then that population isn't inferred to have much climate change vulnerability. So over the last decade, these methods have increased in use and in numbers. But despite their increase in popularity, there are only a handful of cases where folks have tried to verify the predictions from these methods. This is a paper that we published this year that came out in Global Change Biology, and I can't get into it too much, but what we showed with empirical data from 3 conifer taxa is that these gradient forest models do well to predict performance in a common garden, but there are caveats. And I don't have time to enumerate those caveats, but what those caveats add up to is the conclusion that the domain of applicability for these genomic offsets is largely unknown, meaning we do not know the circumstances that they'll do well or the circumstances that they'll do poorly. And so the domain of applicability refers to the circumstances under which a method is acceptably accurate. And for genomic offsets, that domain encompasses the evolutionary history of the targeted populations, as well as the experimental design that is used to train and validate the model itself. One of the reasons that these validation attempts are so few is because the data necessary to validate these models is also rare. But fortunately with simulation data, we can kind of sidestep the weight and the resources and generate data to test these methods in silico. And so with these simulations, we want to create circumstances that represent biological realism, contrasting cases, and adversarial scenarios. So biological realism refers to the simulated data reflecting patterns in empirical data sets like environmental climes. Here I'm showing just sea surface temperature. These simulations should present contrasting cases like varying population connectivity, but maintaining some average population differentiation, and present adversarial scenarios where datasets capture extreme characteristics of empirical data that could challenge these methods, such as predicting to novel climate change scenarios. So with this in mind, we created simulation datasets that tried to capture these three goals. So over the next few slides, I'm going to be explaining the scenarios that we simulated. The first one is the arrangement of environmental data on the landscape. So we simulated 100 populations with at least one or two environments to which the population is adapted. The first landscape is a simple Klein scenario where we have a temperature-like environmental variable that has a north to south Klein, and then a second environmental variable that has a Klein from east to west. We have a mountain scenario where that second environmental Klein represents elevation. We still have that temperature gradient though. And a third scenario which is representative of estuary Kleins. And what we were trying to get at is simulating an environment where there's a marine environment that populations have connectivity, but inlets that have Kleins in salinity. So with these two landscapes on the outside, we have geographic distance corresponds to environmental distance. We might expect these scenarios to perform well with genomic offset methods. But here in the center, we have populations on either end of the landscape that have the same multivariate environment. But of course, even with isolation by distance, we expect the genetic composition to be changed. And so will that present a challenge to methods? So the next few levels of the simulation, I'm going to go by rather quickly because these levels didn't show to have much effect on performance. But we did, like I said, we had one trait, that was the environment that populations adapted to, or in some cases, two traits were adapted to. And in some scenarios, we had equal selection on those two traits. In other scenarios, we had unequal selection strength. And then in other scenarios, we allowed for pleiotropy in the same type of situations. We also varied population size and migration connectivity. In several scenarios, we varied the level of polygenicity, signifying the number of loci that underlied the adaptation to those environments, ranging from a few loci to many thousands. And we tested 5 implementations of 4 genomic offset methods that have become popular in the literature. And what I want you to take from this slide is that 4 of these can take multivariate climate data, while 1 cannot. And 2 of these can allow for population genetic structure correction, while the others don't. For the genetic data that I provided, in the paper I provided the whole data set, all loci. Since it's simulated data, we know the adaptive loci, so I could provide that as well, or the neutral loci. But today I'll just be talking about all or the adaptive loci. And then for the climate data that was provided to the models, we used the adaptive environments only. To quantify the performance of these models, we created a reciprocal transplant design across each population on the landscape. And we projected offset each model to each of those 100 populations' home environments. And then in each in silico common garden, we measured performance as the Kendall's tau relationship between fitness that was known and the offset that was predicted. And so for a well-performing model, it should have a strong negative relationship between fitness and offset, so that high offset indicates low fitness and low offset indicates high fitness. So across all these levels, we evaluated more than 3.9 million common gardens predictions, which encompass the evolutionary history and experimental design to attempt to better fill in this domain of applicability of these genomic offset methods. So with this data set, we'll ask 3 questions for the talk today. There were 6 in the manuscript, but I'll talk about 3 today. The first is, how does the past evolutionary history of the populations affect performance of offset methods? So those are the simulation parameters. Does it matter which genomic markers are used between the all or the adaptive markers? And how well do offset models extrapolate to novel environments outside of the range of environments used in training. So how well can these models predict performance and fitness in increasingly novel climates that we expect to see on the landscape? So for the next few slides, I'm going to present figures very similar to this one. We have performance on the y-axis. Note I've inverted it to create more intuitive relationships with the x-axis. And so for this first question, we looked at those simulation parameters and see how they affected offset methods. And overwhelmingly, it was local adaptation in those populations that drove offset's performance. So populations that had high local adaptation had models that performed better on that landscape. And we do see differences among populations. Among those landscapes that I mentioned earlier. And this slide shows each of those genomic offset methods on the x-axis, but what I want you to take from this is that no offset method had similar accuracy across each of these three landscapes. And so this stepping stone Klein scenario was the one where almost all of the methods performed best, as we might expect, and that mountain scenario presented some challenges, and that estuary Klein scenario presented further challenges as well. So no method was performed uniformly well across all these landscape scenarios. But local adaptation is the primary influence on performance, and some of the landscapes could reduce performance. And so for this figure, I'm showing a scatter plot that is color-coded by the density of the points that are on top of each other. And so we have upwards of 10,000 points on top of each other, and the black is between 1 and 100. Well, I didn't mean to show that so early, but on the y-axis we have the performance of models using the whole data set of markers, and then performance of data sets that used adaptive markers only. For LFM2, it's pretty symmetric along this 1-to-1 line, indicating that that marker set isn't all that impactful. But for gradient forests and RDA that's not corrected for population structure, we do see some advantages of identifying adaptive loci. But while they do provide some advantages, they're not universal or by great margins. So for the last question, we ask, how well do offset methods extrapolate to novel environments outside of the range of environments used in training? So let's consider a scenario where we sampled populations on the landscape for genetic and climate data. And given some forecasts of climate change, we know some of these populations will inhabit novel climate environments that were not in the climate data. So that could be in situ related to climate change, or this could be for a restoration project, trying to transplant individuals into a climate that's not in the model. And so what we've done with this model is we've tried to estimate this contemporary fitness function that relates genetic composition to climate gradient. But there could be biological reasons why the trajectory of this function does not follow the same track in these new environments. There could be biological reasons, like if we model temperate species, but then try to project them to really cold or really warm climates, there's going to be reasons why that model will do poorly for biological reasons. But there could also be methodological reasons that the methods don't perform well either. But in our simulations, the true fitness function follows the same trajectory as it does within these contemporary climate scenarios. And so in the best case scenario, can we still predict fitness in these new environments? And so what I've done is I've created novel climate environment scenarios represented by these blue dots. So this is a principal component analysis, and you can see each of these blue dots is increasingly more distant or novel from the contemporary climate space. So these blue dots are on the x-axis here, so increasing standard deviations, novelty. I've also added a climate center for a point of reference. And what we can see is across data sets that have both low and high local adaptation, we see decreasing performance as those environments become increasingly novel. And unfortunately, for the scenarios that did really well for the contemporary offset estimates, those with high local adaptation, those are also the ones that we see the decline faster in these novel environments and performance. And so we saw that performance declines with increasing climate novelty of these offset methods. And no, I didn't show it we did investigate the marker type, and adaptive markers do provide some advantages, but not always, just like in the contemporary scenarios. So with this last bit of information, we can see that these genomic offset methods are probably okay to use with assisted gene flow, where species are moved within contemporary climates, but not necessarily for assisted migration. With that, I'd like to thank everybody for coming and all the people that helped me with this work. I have QR codes for the 2 papers I mentioned on offsets that I've authored today. They link to my research page. I'll be around for questions later, and I'll talk to you then.
Thank you, Brandon. I guess now we have some time to ask questions of all 3 speakers, either Robert, Conchita, or Brandon. And I guess in the chat, Robert has already done a good job of answering a number of the questions that were posed. But I might ask Robert if there is any point that came out of those questions that he'd particularly like to make to the whole audience.
Sure. Yeah, there was one from April about gene expression in different tissue types. And, we can, it would be great to measure this in more than just leaves. The one that I think would be particularly exciting is meristems, because those are the points for future growth. And we see evidence that they're important in legacy effects, which is another way that we might see asymmetries in responses to precipitation. So having a drought year after a wet year, or plants may be able to make it through a dry year after a wet year better than you would expect, just based on their responses to wet. So
Thank you. Let's see. There's one comment about the creation of a seed bank with a focus on genetic resilience. That would be a good way to deal with some of these climate related issues and for plants. And certainly there is that kind of an effort going on both nationally and internationally, which may become increasingly important in the future as the climate continues to change. There are no specific questions for either Conchita or Brandon, but I think it's pretty easy for any of us to track them down if you want to ask questions of them by getting in touch with them individually. So maybe since we're running a little bit behind here in terms of scheduling, I'll thank the speakers and call this session to a close.